We're continuing our discussion of Unit 3, Immunology and Complement. In the previous presentation, we gave an overview of the immune response, whether it's cellular or humoral, and the different steps involved. Now we're going to specifically talk about immunoglobulins. And humans produce specific proteins or immunoglobulins which can be differentiated based on their size, their biologic function, their biochemical properties, and their serological activity. You must be able to draw and label a single antibody structure. This will include the light chains, the heavy chains, the disulfide bridges, the variable region, and the constant region. We're going to discuss the function of antibody parts, and scientists are kind of weird people, and I don't know why they do the things they do, and we're going to learn about some procedures in blood bank that are really, really odd, and who would know to come up with these things. But for antibodies, the scientists treated the antibody molecule with enzymes to determine the function of the different parts. The different structures were formed based on the enzyme used, and the names of the parts are fragment antigen binding, the FAB, or fragment crystalline, FC. We'll first talk about cleavage of an antibody molecule with the enzyme papain. Papain is known as papaya proteinase, which is extracted from the raw fruit of the papaya plant. This splits the antibody molecule above the two disulfide bonds. An antibody digested by papain yields 3 fragments, 2 fragment antigen binding sites, which can sensitize or coat the antigens, and 1 fragment crystalline, which is the region that determines biological properties of the immunoglobulin. Can it cross the placenta? Can it be present in your fluids? Pepsin cleavage uses pepsin, which is an enzyme that breaks down proteins. This splits the antibody molecule just below the 2 disulfide bonds. An antibody digested by pepsin will yield 2 fragments. It forms 1 FAB2, which consists of 2 antigen binding sites joined together by the disulfide bonds. This is able to bind to 2 antigens, which can result in agglutination. So papain, we have 2 fragment antigen binding sites, but they're totally separate. They can attach to or coat an antigen, but they cannot cause agglutination because they're individuals. But in pepsin cleavage, because we have the 2 antigen binding sites still connected, one can connect to an antigen site on one molecule, the other one on the same antigen but on a different molecule, and eventually we can get agglutination. 1 fragment crystalline, and again, this is the region that determines the biological property of the immunoglobulin. And the FC region is only composed of heavy chain portions. And this is just a nice presentation of pepsin and papain in one slide, and the pieces that are formed. The textbook doesn't go into the other types of immunoglobulins, but I want you to refresh your memory on the different immunoglobulin classes. IgM class antibodies are the largest of all the antibody molecules. It consists of 5 of the basic structural units. It's a pentamer. It has mu heavy chains, which are joined together by a structure known as a J chain, which allows for a lot of flexibility. This accounts for about 5 to 10% of the immunoglobulin pool. We don't need a lot of it because it is so big and effective in doing what it needs to do. Because of the size, it's restricted almost entirely to the intravascular spaces. IgM is one of the most important antibody classes that you will be detecting in immunohematology. IgM fixes complement. It's much more efficient than IgG in the activation of complement and agglutination because it is a pentamer. It is the first antibody to be produced and is of greatest importance in the first few days of a primary immune response to an infecting organism or antigen. It does not cross the placenta, and this will be a very important concept going forward when we start talking about the different blood group antibodies. Many blood group antibodies that are capable of agglutinating antigen-positive red cells suspended in saline in tests performed at 22 degrees centigrade are IgM, and they cause visible agglutination. The best example are ABO antibodies. We'll learn that you form naturally occurring antibodies to ABO antigens you do not possess. There are things in nature that are so similar that this is a case in which we get the cross-reactivity. IgM antibodies are potent agglutinators that activate complement very efficiently. IgG class antibodies are the most abundant of the immunoglobulins in the plasma. They consist of one basic structural unit. It's a Y-shaped molecule that has 2 light chains and 2 gamma heavy chains. And what are the light chains? They're going to be either kappa or lambda. They're produced in response to a wide variety of antigens, including bacteria, viruses, red cells, and white cell alloantigens. They coat organisms to enhance phagocytosis by neutrophils and macrophages. Through its ability to cross the placenta, maternal IgG provides the major line of defense against infection for the first few weeks of a baby's life. For example, if the mom has been immunized to measles, mumps, rubella, these IgG class antibodies can cross the placenta, and when the baby's first born, they will provide protection against those types of infections. It is the predominant antibody produced in the secondary immune response. The serologic behavior and characteristics of IgG antibodies make them one of the most clinically significant in blood banking. Clinically significant is the ability to coat and destroy red cells. Most blood group antigens capable of eliciting an immune response result in the production of IgG antibodies. So again, I want you to think of your red cell. It's just covered with proteins, with antigens, and any one of them can elicit an immune response from the person who lacks them. These antibodies are detected by serological test procedures based on their behavior characteristics, such as their reactivity at 37 degrees, activation of complement, indirect agglutination, and hemolysis. Much of the routine blood banking involves serologic test procedures designed to detect and identify IgG antibodies. Again, this is because they are clinically significant and they have the ability to cause hemolysis of transfused cells if they possess an antigen to which the patient has an antibody. There are 4 subclasses which differ in their heavy chain composition and in some of their characteristics, such as biological activities. I want you to know there's IgG 1 through 4, but at the medtech level, you would need to know the characteristics of the specific subclasses, but as an MLT, you are not. IgA class antibodies are found in secretions such as saliva, tears, colostrum, breast milk, and in nasal, bronchial, and intestinal secretions. IgA is present in large quantities in colostrum and breast milk. This is transferred across the gut mucosa in the neonate and plays an important role in protecting the neonate from infection. It's produced in high concentrations by lymphoid tissues lining the GI, respiratory, and genitourinary tracts. It plays an important role in protection against respiratory, urinary tract, and bowel infections and preventing absorption of potential antigens in the food we eat. It represents 10 to 15% of the total circulatory immunoglobulin pool. In plasma, IgA may exist as a single basic structural unit or as a 2 or 3 basic units joined together. The IgA present in secretions exists as two basic units, a dimer, attached to another molecule known as the secretory component. This substance is produced by the cells lining the mucous membranes. It is thought to protect the IgA in secretions from destruction by digestive enzymes. IgA does not cross the placenta and does not bind complement. For blood banking, an IgA deficient individual may produce anti-IgA, which can cause severe, life-threatening anaphylactic reactions during a blood transfusion. Once these individuals are identified, they must be transfused with blood and components which lack IgA, which is relatively easy because 20% of the population is IgA deficient. The bottom picture shows you the three formations of IgA. IgE is a trace plasma protein, only about 0.004% in the plasma of non-parasitized individuals. It's of major importance in mediating some types of allergic reactions and is generally responsible for an individual's immunity to invading parasites. The Fc region binds strongly to a receptor on mast cells and basophils, and when antigen is bound, it causes the basophil or the mast cell to release histamines and heparin from these cells that result in allergic reactions or symptoms. The clinical effects of IgE-mediated reactions include increased vascular permeability, skin rashes, respiratory tract constriction, which results in wheezing, and increased secretions from the epithelium, watery eyes, runny nose. Not much else is known about its biological role. IgE does not fix complement and does not cross the placenta. No blood group antibodies have been reported to belong to this class. This picture illustrates the role IgE plays in allergic reactions. IgD accounts for less than 1% of the total immunoglobulin pool. This is primarily a cell membrane immunoglobulin found on the surface of B lymphocytes. IgD does not fix complement and does not cross the placenta. Little is known about the function of this class of antibody. No blood group antibodies have been reported to belong to this class. A blood group antibody is considered clinically significant if it has been associated with the following. It has caused hemolytic transfusion reactions, which is the destruction of transfused red cells, or it has been implicated in hemolytic disease of the fetus and newborn, which is the destruction of fetal cells. The mother has formed antigens against clinical significance of blood group antibodies. You must know this and remember this for the rest of your professional life. A blood group antibody is considered clinically significant if it has been associated with the following. If it has caused a hemolytic transfusion reaction, also known as HTR, this is caused by the reaction of a patient antibodies against antigens on donor cells that will result in their destruction. Bad thing to happen. If it's been implicated in hemolytic disease of the fetus and newborn, HDFN, this is caused by the reaction of maternal antibodies, which are directed against fetal red cell antigens that have been inherited by the father. Both of these topics will be discussed in great detail later in this course. The ABO blood group system is the most important blood group system that we will discuss. After ABO, there are at least 34 other blood groups with over 600 antigens. Again, we're talking blood groups. We're talking about antigens that are present on cells. Individuals may produce antibodies to blood group antigens they do not possess when exposed to blood through transfusion or pregnancy. The second exposure to the same antigen may result in immune hemolysis of the red cells. A transfusion reaction is a term used to describe an unfavorable response by a recipient to the infusion of donor blood or blood products, and they include the following: in vivo hemolysis, either immediate, such as in a secondary response, or delayed, which is caused by a primary immune response. It results in decreased survival of transfused cells. Anaphylaxis is another one, usually due to IgA antibodies present in the recipient. Graft versus host disease, post-transfusion purpura, alloimmunization. This is allo. You're developing or have developed antibodies against another human being's antigens. Sepsis due to bacterial contaminated components, and disease transmission. These will be discussed in detail later, but I wanted to give you examples of what transfusion reactions are. They're not always hemolytic. The severity of a transfusion reaction will depend on several factors, including the characteristics of the antibody class involved. Antibodies to the ABO system antigens are predominantly IgM. They cause complement activation, which results in intravascular hemolysis. Intravascular hemolysis is hemolysis occurring within the blood vessels. So intravascular in the blood vessels. Other red cell antigens induce the formation of IgG class antibodies, which may cause accelerated RBC destruction extravascularly. So these are coated with antibody, and they may be phagocytosed, they may be taken out by the spleen. So these are destroyed outside the blood vessels. So intravascular is the worst. Extravascular takes a little bit more time. Symptoms of response to incompatible ABO transfusion may include fever, low back pain, nausea, vomiting, circulatory shock, anemia, jaundice, and kidney failure, which may result in death. Primary immune response may be asymptomatic due to the slow destruction of red cells. So again, in a primary immune response, your body's just learning how to make the antibodies against these antigens. Red cells live 120 days. So by the time there's sufficient antibody present, most of the transfused red cells have died. In a secondary response, it's symptomatic because of the memory cells. They go into the rapid production of antibodies against this specific antigen, which then there will be signs and symptoms of hemolysis in the patient. Antibody-mediated hemolysis can be intravascular or extravascular, and you must know the difference. Intravascular antibodies cause the destruction of the red cells in the circulation. This is always due to IgM and the activation of complement with destruction of the red cells. This is very bad, and you will see red serum or plasma. And the picture on the bottom illustrates the different degrees of hemolysis. As a blood banker, if I spin my sample down and it's a patient who's been recently transfused and I see red, then I'm going to move into high gear because we need to determine what is the cause of the hemolysis. Extravascular hemolysis is due to RBCs being coated with IgG and destroyed outside the circulatory system in the RES system. If it occurs slowly, it may not even be detectable. So it takes an extra step for extravascular hemolysis to occur. Screening donor blood for disease markers has significantly decreased transfusion-transmitted diseases. Reactions to donor white cells and platelets are relatively common, but usually not as severe. ABO reactions are the most severe and the most preventable by following protocols. Other blood group antibodies may or may not be detectable. It is your duty to provide serologically compatible blood and blood components for transfusion. This requires attention to detail, following procedures without question, never skipping steps, never going outside the guidelines of what is entailed by the test. If you do not follow the procedure, then you have the potential for killing a patient. We're going to briefly review complement. Be sure and spend quality time on your notes from serology. Complement is an integral part of the immune system. There's 3 pathways. There's the classical, the alternative or properdin, and the lectin. Complement has 3 primary functions: lysis of antibody-coated cells such as bacteria and red cells, mediation of opsonization, preparation of foreign cells for phagocytosis. Anything coated with complement looks yummy to your phagocytes. Generation of peptide fragments that regulate features of the inflammatory and immune response. The importance in blood banking has 2 major areas. Some antigen-antibody complexes cause sufficient quantities of complement to be bound to the red cells to complete the activation cycle, resulting in hemolysis of transfused cells. Or antigen-antibody complexes initiate complement binding in such a way that allows demonstration of the existence of such complexes using serological techniques. We're talking about in vitro testing, in the tube testing. Fresh serum is necessary to detect complement-mediated in vitro reactions because within 24 to 42 hours in a tube of blood, the complement will be gone. The classic complement pathway involves components numbered C1 to C9. The complement cascade requires the presence of cations, both calcium and magnesium, to be activated. Activation of the classic pathway almost always initiated by immunoglobulin. For IgM, it requires only 1 molecule because there are 5 fragment crystalline components, and that's where the complement will attach. It requires 2 molecules of IgG because they only have 1 Fc component. And again, the IgG must be in close proximity on the red cell for the complement to become seated on the molecules. This illustrates IgG and IgM activation of complement. Again, on the top picture, it requires 2 IgG because the complement will sit on the Fc portions of the molecule. And for IgM, because there's 5 Fc portions, the complement can sit down on 1 molecule and cause the start the activation process. I want you to remember the 3 phases of activation in the classic pathway. That's the recognition phase, the activation phase, and the attack phase. At the end of the story, the classic pathway involves activation of the complement components in this sequence. I want you to memorize this sequence. It must go to completion for hemolysis to occur. The next 2 slides are to assist you in your studies. This shows the different complement components in the classical pathway of activation. And I like this picture because it's a little bit more detailed in showing the actual activation on the cell membrane. The alternative, also known as the properdin pathway, the proteins in the alternative pathway perform activities similar to those in the classic pathway, but they are usually non-antibody triggered. The classic pathway is always triggered by antibody. Any one of a variety of substances can initiate complement activation in the alternative pathway, including bacterial polysaccharides and lipopolysaccharides, endotoxins, cobra venom, trypsin-like enzymes, aggregates of IgA and IgG4 that do not activate C1. In the alternative pathway, C1, C4, C2 do not participate. It starts at C3 and then goes to completion. The third pathway is the lectin pathway, and activation begins when mannan binding protein, MBP, binds to the mannose group of microbial carbohydrates. Two more lectin pathway proteins called MASP1 and MASP2, which are equivalent to C1R and C1S of the classical pathway, now bind to the mannose binding protein. This forms an enzyme similar to C1 of the classical complement pathway, and it's able to cleave C4 and C2 to form the C4b, C2a, the C3 convertase, which is capable of enzymatically splitting hundreds of molecules of C3 into C3a and C3b. The beneficial results are the same as in the classical complement pathway. It triggers inflammation. It chemotactically attracts phagocytes to the infection site. It promotes the attachment of antigens to phagocytes via enhanced attachment or opsonization. It serves as a second signal for activation of naive B lymphocytes. It causes the lysis of gram-negative bacteria and human cells displaying foreign epitopes, and it removes harmful immune complexes from the body. Here is a picture that illustrates a summary of the activation of the pathways. The classical pathway requires antigen-antibody complexes, the lectin binding to pathogen surfaces, and the alternative pathway, pathogen surfaces. And they all end up in the same place. Regulation of complement is very important. The activation of the complement cascades results in a complex series of molecular events with potential biologic consequences. Modulating mechanisms are necessary to regulate complement activation and control production of biologically active split products. The first mechanism is the spontaneous decay of activated components that are not involved. They're split off from the complement, thrown into the plasma, and then just go away. The second mechanism involves specific control proteins that modulate the activity of certain complement components at critical activation steps. C1 inhibitor blocks activities of C1R and C1S. There are other factors that inhibit activation of other complement components. A number of proteins act to control the membrane attack unit. The bottom line is you got to turn it off. As always, review your objectives, read your textbook, and review the presentation as often as necessary. Some references.